but I'll just do a quick intro for those that you're obviously doing your postdoc research at UTS now, so moved from Macquarie University, and
yes,
and your project with us is interesting title, Cage Fighting Against Cancer. So in particular, neuroblastoma, which is the most common solid tumor in childhood cancer. So without any further ado, I'm going to pass over to Andrew, and he's going to give us a bit of a spiel on what he's been doing. Then I'm going to ask him a few questions at the end. And if there's any other questions while we're chatting, you can use the chat bar down the bottom, and then those questions will come up and I can ask Andrew those as well. But Andrew is one of our young and up and coming researchers, and that's one of the things that we love. Like one of the the funding round that we've just started at the Kids Cancer Project, there's no limit on the age or the experience. The track record of the researcher is not something we look at. We look at their ability to look at blue sky and groundbreaking opportunities and make a real difference. So we're looking at sort of innovative and novel treatment options, and sometimes it doesn't, anyone can come up with those ideas, and Andrew's got one of them, and I'm sure he'll share some of that science with us now. So thanks for joining us, Dr. Andrew Kerr.
Thanks, Owen, and hello to everyone. It's great to be here, and thank you especially to the Kids Cancer Project for supporting my project and the work that we're doing in my lab. And I hope today, I can show a few of the things that we're doing and some of the things we want to do. And I hope you enjoy the innovation we're trying to make. So I'll just try to share my screen. How does that look to you guys? Is that in presentation mode on?
Yeah, it is. You've just flipped it over so we can see it. It's just your first page up at the moment.
Okay, fantastic. Great. So I'm going to be talking about cage fighting with cancer. And so this is, I'll run through a bit of the stuff we're doing at UTS, where I'm based now. I've just moved from Macquarie to UTS. And we specialize in combining the areas of synthetic biology and nanomedicine for the treatment of diseases, with a lot of focus in the area of cancer. So just a little run through of nanomedicine if you're not familiar with it. So nanomedicine is a very multidisciplinary field, and we're trying to make breakthroughs in treatments and diagnostics using things that are constructed at the nanometer scale. So to give you a bit of a perspective on that, if you took a 1 nanometer marble, relative size, 1 nanometer compared to 1 meter would be the equivalent of a marble to the size of the Earth. So 1 nanometer is 1/1000th of 1 meter. So we're working at a very, very, very small kind of scale. So in nanomedicine, the key component is nanoparticles. So you can see we're working in this 1 to 100 nanometer area here. And nanoparticles can be made out of lots of different components. They can have lots of different forms and properties. So you can have something that's made out of metal, from lipids, gold particles, which might have different optical properties, quantum dots, which are semiconducting materials that are fluorescent in different color ranges. You can have silica particles with iron loaded inside them that can be used for magnetic resonance imaging. And then you can have the bacterial protein cages, which I'm going to talk about today, and they're all made out of proteins. So why use nanoparticles, and why use them for drug delivery in the treatment of cancer? So nanoparticles can be loaded with different types of drugs, so it allows you to do different types of therapy at the same time. Nanoparticles allow you to deliver different ways into the body. You can inject them, or you can inhale them, for example. They improve the stability of a drug and the therapeutic potency of a drug. They can also extend and improve their pharmacokinetics, so prolong their blood circulation or reduce any side effects. And one important thing about them is that you can also attach things to the outside that allow you to target that nanoparticle to somewhere in the body. So that allows targeted therapies. So some of the clinically approved nanoparticles that are out there now are just shown here. So we've got something like Gardasil, which is a protein nanoparticle that's basically a vaccine that prevents the cervical cancer. You can have femoxytol here, which is a an iron oxide, a polymer-coated iron oxide particle that allows you to image MRI in the brain. We've got here another protein particle that allows you, that's a chemotherapy drug. So there's lots of things that are available, but unfortunately, there's not as many as that we would like to have out there. There's not as many nanomedicines that have gone from the bench, the lab bench, into the clinic. So there's a lot of effort going into finding new and innovative ways to actually start translating nanomedicines into the clinic. So what we're interested in is a kind of a different type of nanoparticle that might have potential for the clinic. And this is where the innovation comes in. We're really interested in figuring out whether we can use protein nanocages to treat disease. So these are protein nanocages. They're made out of proteins. They self-assemble to form these nanoparticle structures, like imagine like a hollow soccer ball or something. And the unique thing about protein nanocages is they're made inside cells. So we genetically engineer bacteria to make them. And that means we can also engineer the bacteria to make them to have different functions. So we can genetically engineer them to do different things. And so the protein nanocage we're interested in is called encapsulin. And these are actually found naturally inside bacteria. And inside bacteria, they form these little encapsulin, little nanoparticles here made out of protein that are hollow. And they actually normally encapsulate a protein. So they have this unique system here that allows you to, that has a cargo protein, like an enzyme, for example, and it selectively recognizes this enzyme that has a little tag on it, and it'll self-assemble around, the nanoparticle protein will assemble around it, forming a protein-loaded nanoparticle. So you've got this natural system where you can naturally load in proteins into a nanoparticle, all inside a cell, which we then can take out and use. So I'll just skip ahead here. So this is kind of the concept I'm talking about with encapsulins that's unique, is that we can genetically engineer a cell to have a protein. So instead of the natural enzyme, here we've got a fluorescent protein called green fluorescent protein, GFP. We've engineered it to have this little tag on it, and then we've produced it inside the cell with the component of the nanocage. That all comes together and assembles and creates a fluorescent protein-loaded nanocage. So here, if we look over here, we can see our bacteria glow in the dark because they've got this glow-in-the-dark cage inside them. We take that cage out, and we can see under an electron microscope that we get this protein-loaded cage. Here, they're all nice and circular, and inside there, you can see the protein. And when you shine a blue light, they fluoresce because it's a glow-in-the-dark protein. So there we go. This is a plug-and-play system that we want to use to start loading therapeutic proteins inside encapsulin using this genetic engineering plug-and-play approach. So I'll just run through one thing that we've recently published to show where we're at, and then I'll show what we're doing with neuroblastoma. So we recently published a paper around reprogramming these encapsulins for photodynamic therapy. So what this involved was we took a protein that when you shine light on it, it converts normal oxygen inside a cell into reactive oxygen, or ROS, and that's toxic to the cell. So we take these cages, we genetically engineer them to have this protein, we shine a light on them, they do this conversion, and we can show that we can generate this ROS when we shine a laser on them. And then the idea in PDT is to take this into PDT. In PDT, you have a fluorescent drug called a photosensitizer, and you activate it with light to do that conversion process of normal oxygen to reactive oxygen. When you do that inside a cell, you kill it. So what we've done is we've got our nanocage that we just showed just then, where we can shine a blue light on it. What we do is we deliver that into the tumor cells. We shine a light on the tumor cells. Those cages then start converting the oxygen in that tumor to reactive oxygen species and kill the cells. So I'll just show some quick results here. So in the dark, when we mix our cages, when they're empty, they don't have any protein inside, they're just an empty cage, and we mix them with tumor cells. I'll just show you this light. If you look here, there's no change, there's no dip here, it's all straight. That's showing that when we incubate our empty cage with these cancer cells, they have no negative effect on them. They don't kill any cells. When we add light, they still don't kill any cells. That line doesn't change. On the other hand, when we do have our protein cage here, and we mix it with these particles, this is the green dots I'm talking about now, there's no change in the dark. So the cells these cancer cells we mix them with aren't getting killed. But then when we do add light, you can see this green line again shows that it dips in the viability of these tumor cells down to about 60%. So that's saying that when we put these in, they go into the cells. When we shine the light, they start killing the cells. And after 12 hours, it's destroying about 50% of the cell's viability there. So this is the first time that encapsulants had been shown to encapsulate a therapeutic-like protein, and we've delivered it into a tumor cell, and we've used light-activated therapy to kill those cells. So it was kind of a really novel thing. We're quite proud of that work. And the funding from Kids Cancer Project actually helped us finish this off. And we just got it across the line early this year. And you can read the paper that's out in Applied Materials and Interfaces. So a bit of fun work there as well. Like, it's quite interesting. It just shows that this platform's like really unique, and we can really just play around. There's lots of opportunities there. So moving into neuroblastoma. Like Owen gave a bit of a good introduction to neuroblastoma. So it is the most common extracranial solid tumor in young children. After brain cancer, it's the most lethal type. And at the moment, a lot of the treatments, the treatments are really harsh. They have a lot of side effects, and they have these long-term negative effects in those patients that do survive. So interesting enough, through our collaborator, Dr. Orazio Vittorio at Children's Cancer Institute, I stumbled across that neuroblastomas apparently have high levels of copper and makes them susceptible to copper-based therapies. So you can lower the copper amount, or you can increase the copper amount to disrupt that homeostasis and maybe kill those neuroblastoma cells. So the concept was that perhaps we could then engineer these encapsulants to encapsulate a protein that allows us to suck in copper, like load the cage with copper, which then we can deliver into neuroblastoma cells to overwhelm that high copper content to a point where it kills those neuroblastoma cells. And that process, we deliver too much copper, is called copper ionic therapy. So this is just a diagram showing that kind of concept where we have a cage that is able to take in copper. We then deliver a copper-loaded cage into the neuroblastoma cell, where there's already high levels of copper. This extra copper causes the cell to stress out, and it causes oxidative stress that causes it to go into programmed cell death, leading to death of the tumor. So basically engineering a cage to deliver copper into neuroblastoma. So this is where we kind of are now. We've bioengineered encapsulant to encapsulate these different copper storage proteins that we found from a bacteria that binds lots of copper. And we loaded these, we did the same concept as I showed with the photosensitizing protein before, is we took our encapsulant, we took a cell, we engineered it to produce an encapsulant nanocage that has a copper storage protein inside it now. We add copper to that, and it would sequester, suck in the copper and load the copper inside the cage, which it doesn't do naturally. So if we look here, this is some electron microscopy that we've done, showing that we can load copper in using this system. So we have an empty cage here, so nothing should go inside, there's no protein. And you'll see here, in this here, when we add no copper, we don't see anything here. There's no electron dense dots showing us that there's any copper dots, copper accumulation in the sample. And even when we add copper, we still don't really see anything. So this is just showing us that there's no copper going into these cages when there's no protein. On the other hand, if we just look at this one here, this is a copper storage protein 1 put inside the cage, so one of these. When we add copper, you see these big black dots starting to come in under the electron microscope. And that's telling us that yes, we've made a cage that can now suck in copper and accumulate it inside. So yeah, we've got these copper sequestering, copper loaded nanocages. So the next part of what we're running in parallel is like, okay, so can we actually target neuroblastoma with these cages? We know we can load them with copper now, but can we target neuroblastoma specifically? If we can do that, then we reduce those off-target side effects inside a patient. So what we did is we took our nanocage and we genetically modified the outside of it to display these little peptides, little protein sequences that bind to integrins, which are overexpressed on the surface of neuroblastoma. So we take the cage that doesn't have anything on the surface, and we mix it with neuroblastoma cells. And we've fluorescently labeled the cages, so there's no fluorescence here. But when we compare that to cages that we've genetically engineered to have that peptide on the outside, we can see these little green dots here, which are dye-labeled nanocages going inside the neuroblastoma cells. So this is telling us when we modify those cages with this peptide, they can selectively target and enter into the neuroblastoma cells. So we've modified them for targeted, potentially for some targeted delivery of those cages. So we've got the two steps there. We've got the ability to load these cages with copper now, and we've also got another ability to potentially deliver those into neuroblastoma. So we've done a little bit of preclinical work just to look at like how encapsulants would interact with the body. We've had a few disruptions due to COVID, but at the moment we've got some preliminary data here. So what we've done is we've taken our nano cages and we wanted to see if they were safe when we put them inside an animal. And we also wanted to see whereabouts they go inside the body, which will help us to then improve the targeting again. So we administered encapsulants that we labeled with a special dye, and we injected them into mice that we modified to have a neuroblastoma tumor growing on them. So a neuroblastoma tumor bearing mouse model. So we injected the fluorescent encapsulant in, and then we monitored where it went in the body over 24 hours. So if we just look here, this is the free dye itself. You can see that it goes throughout the body and then it's rapidly cleared from the body. It doesn't accumulate anywhere. A little bit in the kidneys, and then it leaves the body within 24 hours. Even after 7, it's mostly gone. However, when you have the encapsulant on there, the encapsulant with the dye, you can see that it stays in the body a bit longer and it accumulates here in the liver instead. And after 24 hours, you start seeing more clearance. So this is telling us that the body's immune system's recognizing encapsulant, and it's pushing it, it's clearing it through the immune system via the liver. But what we know is that it's safe to administer to animals, and we didn't see any toxic effects when we injected them in. And now we know where they're going in the body and what we need to do to modify them to better target them into tumors. So the next steps are going to be about understanding those interactions between encapsulants in the body and improve that in vivo targeting, maybe putting something on the outside that masks it from the immune system for a bit longer. We also want to start looking at whether we can, just in a cell culture, administer copper ionic therapy with our copper-loaded protein cages. And then if those are running in parallel, hopefully we can get to a point where then we can start putting our copper-loaded cages into a neuroblastoma model and start reducing those neuroblastoma tumors as a treatment option. So just thanks to everyone in our research group. A lot of this work's been done by my PhD student, Sandra, out at Macquarie. And especially thanks to Dr. Orazio Vittorio from Children's Cancer Institute, who helped me come up with this whole idea. So thanks. And again, thank you again to Kids Cancer Project for funding.
Thanks, Andrew. And thanks for sharing your screen with us and giving us a wonderful opportunity. So if anyone's got any questions, you can just enter those through the chat. But I had a couple to start with. Obviously, for you, Andrew, as a young researcher, one of the focuses of the Kids Cancer Project, as I said at the start, is not having age and background and track record as an important component. How, for us, that then gives us an opportunity to future-proof the next generation of childhood cancer research. As a young researcher, early researcher, how important is some of this funding to get you started in proof of concepts and gather data on this information that you've got to date?
Oh, I think the best way to say is game changer. Like, I wouldn't be able to do any of this without the support from the Cancer Australia Kids Cancer Project grant. It's really kind of transformed our capacity to do the research that we want to do. And it's also opened a lot of doors. Like, it gives more opportunities. There's a knock-on effect from having that funding that comes with it, that also helps progress the early career researcher. So yeah, really important.
Yeah, it is. I mean, one of the scheme that Andrew's talking about, for those that we've spoken about it, obviously at the Kids Cancer Project, but is the Cancer Australia PDCCRS, so the Priority Driven Collaborative Cancer Research Scheme. So even for us, it gives us an opportunity to partner with other bodies, other funding bodies. So, with Cancer Australia and Neuroblastoma and Cure Brain Cancer and all these other charities. So it gives the opportunity of like-minded organisations to come together because, as you said, collaborative, research and sharing. So obviously, you work with CCI and, overdosing copper and, copper being a key to neuroblastoma and your protein nanocages and delivery. So what sort of pipeline, how long has this sort of science been in process, just to give us an idea of, conceptually when the first components of this came to you? So, because it's something you're leading as part of your team, isn't it?
Yeah. So I've been kind of developing this kind of technologies over probably the last 4 years. So my background was originally protein engineering. And then I was quite interested in engineering proteins to put on the outside of nanoparticles to help target them somewhere. And then I kind of combined those things to have, protein particles. And then to kick that off about 3 years ago, I got a Cancer Institute New South Wales fellowship. And that's where that photodynamic therapy project kind of started off and started coming together. So over the last 2 or 3 years ago is where I started the whole concept of these cages and engineering them. And then, it's kind of led me up to all the techniques and technical stuff that we've got now has led to where we are with these projects.
So then obviously the photodynamics in the lab is what you're bringing next. So how would that be used in the clinic, for example? So if the nano cages were delivered and they were full of copper, how would the light be? So how would that clinically, how do you see that moving clinically? Would it be an external light source, or how does that work in the human body?
Yeah, so it's good to put out. I probably didn't make that too clear. I was probably rushing a bit. So they're two different technologies. They're two separate things. So one's photodynamic therapy with a different type of protein, and the other one's the copper, which is you're delivering something else. So they're two different treatment modalities. So it depends on what we're trying to treat. So for example, when we're doing the light therapy with the PDT, it might be more suited towards something. For example, we did it with lung cancers, because you can modify bronchoscopes that go inside the lungs and you can modify them to deliver light to that area. So it does depend on the modality and how you activate the treatment. But as you can see with the copper treatment, we're trying to deliver it specifically, like you would inject it into the bloodstream, and then we were hoping that it would accumulate in the tumors where it would start delivering and go into the cells and deliver the copper there. So two different kind of modalities,
Okay.
two different treatments,
So it's obviously very complex too. You said that cross multidisciplinary, because you've got genetic engineering and biologically derived nano cages. You're looking at different nanotechnologies, nanomedicine. It's a rather large project. So the importance of your collaboration with other members of your team and other institutions outside CCI.
So I was quite lucky. So when I finished my PhD about 5 years ago, I was lucky enough to be part of the Center for Nanoscale Biophotonics, which is a really multidisciplinary center that brings together biologists, chemists, and physicists into one room. And there's about 200 researchers in this center. And that's kind of where all that, just talking to people outside your field, trying to get a common language really became important. But then it also allowed to do stuff like this, where you're thinking really innovative and outside the box. That's kind of, I had a background of getting into the multidisciplinary quite early. So I guess that's why you can see that there's quite a lot of moving parts going on.
Yeah, that's great. So that's it, Andrew. I don't think we've got any questions from anyone else, but I'd just like to see say congratulations, obviously, on your research. The Kids Cancer Project are really proud to be able to fund you. Just, even on this webinar, the excitement in your voice and the smile on your face, and and for us, the difference this can possibly make for kids down the track is very exciting. The collaboration with other sort of institutions and looking at a target like neuroblastoma. So I had one of my Wallaby teammates. We both had 2-year-old children, and his 2-year-old son was diagnosed with stage 4 neuroblastoma. Both his son and my daughter finished their HSC last year, so it's exciting to see what science can do. But also then, for me, it's the most important thing is that the thing that you said is like reducing current treatments and the toxicity and the side effects of that. So it's something the Kids Cancer Project are really passionate about. And as we said beforehand, we're launching our Better Challenge, which is encouraging people to run, walk, or ride 90 kilometres across September as part of Childhood Cancer Awareness Month. And that's the concept, because every day 3 people are getting diagnosed. And if we can fund bold, innovative scientists like you to look at innovative and novel treatments, they're going to reduce the side effects for these kids with cancers. It's amazing. So it's something Cole Reynolds started 26 years ago. And if he was online now with you, he'd probably be trying to give you a big hug or a big clap. But on behalf of the Kids Cancer Project, Andrew, thank you very much for your time today. And I'm sure you've left everyone a bit more insightful on cage fighting in cancer. So thank you.
Thank you, Owen, and thanks to the Kids Cancer Project. It's much appreciated.
Cheers, mate. And if you get a chance, ask those All Blacks to take it easy on the Wallabies next time they play.
I'll try my best.
Okay. All right. Thanks all. Thank you for joining us for, I think our next Behind the Science will be in a couple of months, and look forward to delving deeper into another one of the Kids Cancer Project's 32 research projects this financial year. So thank you. Thank you all for joining us. Thanks, Andrew.
Thanks. Bye.